Hello, my name is Kelly Schmidt, and today I'm going to teach you about genetics. The importance of genes. It is a basic unit of variety in a living organism. Our genes come from our parents, so one from your mom and one from your dad, a gene from mom and a gene from dad. You may inherit physical traits and certain diseases and conditions from a parent. So just because, let's say, mom has diabetes, but dad doesn't, that doesn't mean you will get diabetes. There's a percent of a chance that you might get it, but you might not have it, but then maybe, let's say, a sibling might get that diabetes effect. Also, it contains the data that we need to build and maintain cells and pass genetic information to an offspring. Mitosis versus meiosis. Mitosis has 4 stages. It is prophase, metaphase, anaphase, and telophase. What my biology teacher in high school told me was to remember as PMAT. PMAT, prophase, metaphase, anaphase, telophase. So mitosis happens within a somatic cell. It produces 2 diploid daughter cells. The chromosome number remains the same, so it will start with 46 and end with 46 in each cell because it produces 2 daughter cells. The genetic variation will not change. They produce new cells, and they start with a single parent cell. So they start with 1 and end with 2, and it takes about 20 minutes. Whereas meiosis has a total of 8 stages, so that's PMAT twice, prophase twice, metaphase done twice, and telophase done twice. They happen within germ cells, whereas remember mitosis happened with a somatic cell. The purpose of sexual reproduction. Remember, the purpose of mitosis was cellular respiration. They produce 4 haploid cells. So in mitosis, they produce 2 diploid cells, where in meiosis, they'll produce 4 haploid cells. The chromosome number is half upon each daughter cell. So before, it was only 2 daughter cells. Now here, it's half the daughter cells. There's 2, so there's 4 of them that will remain. There's 4 that will be there. The genetic variation in meiosis is increased, where remember in mitosis, they did not change. And they start with a single parent cell, just like mitosis did, and they produce new cells just like mitosis did. So in fruit flies, it takes about 1 to 2 days, where in a human male cell, it will take up to 4 days. So see in this picture, not sure if you can see it right well, but it says start. Mitosis starts with 46 diploid chromosomes, then ends with 2 diploid cells that both have 46 chromosomes. So the chromosome number stays the same. Where in meiosis, it starts the diploid cell with 46 chromosomes, and then they produce 4 haploid cells, and the chromosomes number is halved, so that is 23. 23 in one, 23 in the other, 23 in the other one, and 23 in the other one. So 4 haploid cells that have 23 chromosomes in each cell. Next, we have DNA versus RNA. DNA is a double helix, so it is double-stranded, and the sugar that it uses is deoxyribose. It has 4 bases: adenine, guanine, thymine, and cytosine. It is a phosphate and a nucleic acid, whereas RNA is single-stranded, so it's not a double helix. It has its own strand, and it is used with ribose sugar. It also has 3 bases. It has adenine, guanine, cytosine, and uracil. Instead of thymine, RNA uses uracil. And it is also a phosphate and a nucleic acid. In this picture right here, it shows what adenine looks like, what guanine looks like, what cytosine looks like, and thymine looks like for DNA. And then for RNA, it shows adenine, guanine, cytosine, and uracil. So see in DNA how it has a big old backbend because it's doubled. So because it's a double helix, so it's doubled on each side. It's exactly the same. And for RNA, it's singled because it's a single-stranded. The cell cycle. It is a series of events that take place it is a series of events that takes place in a cell as it grows and divides. It has 4 phases: G1, which is our growth stage, S, which is our synthesis stage, G2, which is the growth and prepare for mitosis, and M, which is the mitosis stage, which is cell division. So remember, it will produce 2 diploid cells that each have 46 chromosomes. It will start with 46 and end with 46, but it will have 2. So one from mom and one from dad. 2 chromosomes, both with 46 chromosomes. Genetic coding is the term we use for the way that the 4 bases of DNA, the adenine, cytosine, guanine, and thymines, are strung together. So it will create a long chain of amino acids. Cytosine is C, guanine is used with G, thymine is used with A, adenine is used with adenine is used with A, thymine is used with T, and U is used with uracil. So C pairs with G, so cytosine with guanine, G pairs with C, guanine with cytosine, A pairs with U, adenine and uracil, and T pairs with A, thymine and adenine. Remember, there is no T, there's no thymine in RNA. Instead of thymine, we have uracil. So uracil is paired with adenine because DNA has all of those the same as RNA except one. T is replaced with U. And then I have, and then right here we have the code of genetic coding. So you start with your first letter. So say we have a code that is UCA. So we start with this one. So it says first letter. So you start here, you find U, and you keep going till you get the second letter. So our second letter is C. And then our third letter is A, UCA. So that is, the genetic code is SER, S-E-R. But then say we had UUA, we would start with U, then we go to UUA, and it says stop. So that's where, if you run over a stop, it means that your amino acid chain has ended. That's the end of it if it says stop. And even if you have more letters, you do not keep going, you stop there. And then I have these 2 videos on mitosis versus meiosis, and then RNA versus DNA. The first video is 2 minutes and 42 seconds long, and then the second video is 4 minutes and 43 seconds. So RNA and DNA is 4 minutes and 43 seconds, and the meiosis is 2 minutes and 42 seconds. Thank you.